Okay. So first of all, thank you so much to Georgia. I'm used to speaking in such different setups, usually, science oriented only talk. So I was actually really excited to make this talk. I probably spent way too much time, looking for different things to add to my presentation. And because I think we're a relatively low number of people, if this was a conference room, I would say come to the front, everyone. Let's have it as like a casual discussion. Please feel free to stop me at any time. What I'm going to do is I'm going to make this small. So unfortunately, I will not see your faces because otherwise I will not see my screen. But raise your hand or unmute yourself to ask questions. Like I said, this is my first time presenting it in such a context or such format. So hopefully you like what I prepared. Okay, so the outline is fairly simple. I'm going to talk a little bit about myself, even though Georgia kind of like gave already most of it, but sort of from my point of view, I'll talk a little bit about my lab's research. It might be more like 15 minutes, so I apologize, but hang in there. I will relate to some advice regarding graduate school. I actually, as Georgia mentioned, met her through her interest in the Transstitutional Program in Chemical Biology, which is the graduate program I draw most of my students from. The Tri-Institute includes Weill Cornell Medicine, Sloan Kettering, Cancer Center, and Rockefeller University. And together, there are about 55 labs that do different kinds of chemical biology. So I've seen everyone from, synthetic, inorganic, all the way to, through biophysical, structural, and even cancer biologists that are interested in doing interdisciplinary research. So I think I can relate to many different backgrounds. So hopefully I can give advice that is general enough. And then I was discussing with Georgia yesterday about random things people usually ask me when I give career advice and do career tables or career panels at meetings. So I made a few slides at the end about that, and hopefully we'll also have enough time to discuss new questions. And again, feel free to interrupt at any time. Okay, so I was, as Georgia sort of mentioned, I started my career in the US, but I was actually born in Israel in an underscore prior to 2000. I started my BSc in biology, actually at Tel Aviv University, but then transferred to SUNY Stony Brook. Parents moved to the States. I then moved back to Israel to do my PhD at the Weizmann Institute, and moved back to New Jersey to do my postdoc with Tom Muir, who's a very chemical biologist. And I started my lab at Memorial Sloan Kettering Cancer, just across the river in Manhattan, September 2016. Now, all these, moving across ocean sound like a pretty, Israel and the East Coast is about 5,012 miles apart. And I've moved 3 times, even though that include visits to the family. So it's about 1710 to the third in terms of scale. But I always transition I did in science is far more extensive, because I started as a neurobiologist doing animal models as an undergrad, as a postdoc in cell biology, did my PhD in biochemistry, postdoc in chemical biology, going from a 10 centimeter animal 0.3 to angstrom, that's about 75 10 to the 9th. So definitely a bigger transition in my career. My lab is primarily interested in understanding fundamental mechanisms related to epigenetic regulation of transcription, how a misregulation of these events can lead to disease states. Now, as a biologist, we take a very interdisciplinary approach that allow us to really deep mechanistic understanding, linking specific proteins and particularly modifications on histones and transcriptional output. Now, the way we approach or use biology is in two main ways. One is really develop and apply powerful tools that allow us to investigate new biology and sometimes old biology with a new lens, or we study basically chemical events that happen in cells and can change their state. And in this sort of bridge, we go between chemistry and biology, between application and discovery, basic and translational, and even academia and industry, a collaboration. I'll briefly mention one of them today. So, interesting cartoon when I was for other random things, and I felt like I could relate so much to it. A plot of your research topics, because I'm roughly around here, somewhere between here and here. I'm in transitioning to associate professor, and my scientific interest has been growing exponentially. That I can totally see how very soon it will be crazy things happen. But I would say generally, my lab is interested in this physiological relevant form of the DNA in eukaryotic cells, which is chromatin. Chromatin is how each of our cells pack about 2 meters of DNA, which is 6 feet, into a 10 micrometer space. And the way it is conducted is by these structural proteins called histones that are able to sort of package or spool the DNA in a way that allows higher order compaction to fit all this thin thread in the nucleus. But beyond just being structural elements, histones turn out to be a very important and dynamic signaling platforms, mainly through post-translation modifications. Now, my lab is interested in identifying new chemical modifications that occur in histone and can change chromatin structure and function. We're also interested in another family member of the core histones called linker histone that docks to each of these repeating units. This is a mononucleosomes. So each of these repeating units of chromatin and helps compact it. We're also interested in studying much larger modifications such as ubiquitination, and even go into how epigenetic actually regulates viral infection. And we use hepatitis B as a case study. We have a new project that I'm very excited about that is related to epigenetics and immunology. And sort of underlying, we always have this, goal to expand the language of chemical biology for the scientific community by developing probes and doing protein engineering methods to really get a new perspective into the questions that we're interested in. So I will give a few slides about our work on non-enzymatic or chemical modifications, and I'll talk a little bit about our protein engineering, and that will be the whole span of the scientific part of this talk. So the main, biological driving or biological problem that was driving my interest in as an independent scientist is the known link or epidemiological link between changes in our environment, in our nutrition, in our exposure to toxins and drugs, and diseases such as cancer. And as a young, naive, postdoc applying for faculty positions, I proposed the following provocative model where there's actually a direct link between these damaging agents and cell fate by directly damaging chromatin. And this is sort of my hypothesis, was that these modifications change how proteins can dock to chromatin, how the 3D structure of chromatin is affected. And people were very, I would say, skeptical at best. And they were like, oh, this is a crazy hypothesis. And in fact, I had to write a more conservative proposal for my job hunt. And for most places, I submitted both to show I have an innovative side, but also a conservative side that I know there are some things I can do and they're, safer. The funny thing is that I ended up publishing about 20 papers about this really provocative idea. And the first paper from my conservative proposal we submitted last week after years. So sometimes it shows you that being brave doesn't mean not being practical. Okay, so this is, like I said, a axis, but as chemical biologists, we're also interested in the probes to understand this biology, as well as identify vulnerabilities in these process and develop new drugs to target them. And like I said, on a mechanistic level, this all started from the notion that what happens in normal cells, that chromatin is not immersed in PBS or in water or in just simple aqueous buffer, but in fact immersed in a very chemically complex environment, has many reactive molecules that can constantly modify. And it could be chromatin that is open and accessible and described, or chromatin that is packaged and is more structural. And what happens in diseases such as cancer is that metabolism, so it generates more diverse chemical environment and many times reactive chemical environment. And that can change the way certain regions that are supposed to be suppressed, such as that are now being open and expressing and can drive oncogenic programs, and vice versa. Regions are supposed to be open and transcribed, such as tumor suppressors, are now repressed. So the link between the changes in the metabolic and chemical environment and chromatin transcription, where our interest lies. One of the first chemical reactions we decided to look at was glycation. Glycation is something that we all encounter all the time. Creme brulee for super fancy, or in the browning of our steak. It's also the reason why we have wrinkles when we age, because actually the glycation of this extracellular matrix causes cross-linking and decreases the dynamics of our skin, the quality of the skin. And in fact, it's also a very biomarker, because it is the glycation of our hemoglobin is used for diagnosis of diabetes. So in fact, when you get your A1C is the level of glycation of your hemoglobin. Now, glycation is very cool on creme brulee, but how from creme brulee to histones and to chromatin? And the way you get to it is that histones are potentially targets for these chemical reactions, because they have very long half-lives. They actually through cell division, and they are very abundant. And it's told you that they have really important role in maintaining cell fate post-translational modifications. And glycation is a modification on these proteins. So for the real chemists in the audience, I'll just remind you that glycation is the reaction in the case of proteins between nucleophilic side chains such as lysines and arginines, and the open chain form of reducing sugars such as glucose that can form this intermediate shift base that is not very stable, but can rearrange to these amidori products that are more stable. But in my case, I was actually more interested in more downstream metabolic byproducts such as methyl glyoxal that exist at very low levels in normal cells, but at very high levels in cancer cells. And MGO or methyl glyoxal can react readily also with lysines and arginines and rearrange similarly to form these advanced glycation end products. Advanced glycation end products are properly named because they are irreversible. And their acronym is AGEs, and they're associated with age, so that's even more appropriate. These all cause everything that I was telling you about: the creme brulee, the steak, the wrinkles, and your A1C levels. And one of the most important types of AGEs are these cross-linking. Now, cells evolve various mechanisms to fight it. One of them is DJ1, which I'll have a couple of slides about in a few seconds. Also have other mechanisms such as FN3K, both reversing the glycation. It also have mechanisms upstream to prevent the accumulation of methyl glyoxal. But like I said, we've been focusing a lot on methyl glyoxal and DJ1. But we do have a paper about FN3K that was published last year now, if you want to read a little bit more about it. Okay, so in order to understand how these glycation happen and how they accumulate in cells, we developed a series of probes. For methyl glyoxal, we did AlChem-GO, for ribose, azidoribose. And for glyoxal, we just developed this new way to probe it. But we're also developed chemical tools that will help us analyze, for example, inhibition of DJ1. We also developed a series of potential inhibitors using this assay, as well as try to use this methodology to improve CAR-Ts. And what we've discovered through 5 years of research is that chromatin actually undergoes glycation, and that changes its landscape. DJ1 can reverse some of these changes, but some of these more advanced glycation end products are irreversible or more readily or more difficult to reverse. And we show it competes with enzymatic modification, which is another important mechanism, how it disrupts chromatin. And we also found another interesting eraser for these modifications called PAD4, that beyond being an eraser is actually a rewriter. So it removes glycation and adds instead citrullination, or converts the arginine to citrulline. And this is a really interesting, intersection between sugar metabolism, methionine metabolism, and calcium, because PAD4 is a calcium dependent enzyme. Just one word about, for the crowd, about our high throughput assay. It's a recent publication from the lab, where Igor, a really incredible graduate student, PCB graduate student, developed a fluorescent-based assay to identify and in new inhibitors for DJ1 in the screen. We collaborated with the Therapeutics Discovery Institute at Sloan Kettering to identify these new inhibitors. That's another really incredible moiety that the tri-institutional had to do what we call bench to bedside. So if we discover something new that could potentially become a drug, they sort of do the next step and convert it into a drug. We also, in an attempt to identify new scaffolds for inhibitions, we collaborated with near London to do a fragment-based screen, which also gave us some new potential hits, as well as collaborated with InterX, which is a molecular dynamic simulation company that takes the crystal structure of your protein and uses a very sophisticated combination between medicinal chemistry, rational design, and docking to identify potential small molecules that covalently or non-covalently will bind to your protein, and some of it really act as inhibitors. Okay, so I'll just use a couple more minutes to talk about our protein engineering, because this is sort of our bread and butter. And this also from my postdoc, where I was interested in engineering histone cells. And the way I approached it was using something that pretty much only chemical biologists know about, but it's a very, very useful. These are proteins called inteins, and what they do is they bind very, very tightly, and they self-excise, leaving behind them a native polypeptide bond. This is an event called splicing. And what we did is we used this two-component system to test a histone with a fusion to an intine, and then intine carrying a cargo, and we delivered it into cells, allowed the splicing to occur in live cells, making basically synthetic histones in live cells. And in my lab, similar technology to take it one step further and actually manipulate proteins in vivo. So that's really taking chemistry into biology. One approach is to actually do intine splicing in mice brain, and the other one is modifying surface that are then injected into the animal, for example, on CAR Ts. Okay, so this is the entire sort of science heavy, and now I just want to introduce you to my incredible lab and show you how a chemical biology lab is composed. In our case, it's very diverse in every aspect, and scientific background is also that. You can see there are people who do medicinal, organic chemistry, structural biology, immunology, cancer biology, etc. And together, they're really able to tackle these convoluted problems and questions using a very skill set and tool set. I wanted to say a few words about about a PhD. That sometimes when we plan our PhD, we see this flag at the end. We're like, okay, in 5 years we're going to have a PhD, but in reality, PhD is a very, very complex path, and there are many bumps in the road. The highs are very highs, and the lows could be very low. And it's important to know that in advance, because then you're less surprised and discouraged when there are these bumps, and you know that they will also pass. And so I tell my students, PhD, saying I'm done with this paper, done with this PhD, it's rarely the case, because there's always, one more experiment and I'll be done. And they say the last 3 years of your PhD, and I say probably your entire PhD. But that's great, because that's what the reason why we do this research is, because curiosity demands. So we're never really satisfied with what we have. We always want to learn a little more, and that's a great driving force. I wanted to say a word, again, I can expand about each of these topics, but I wanted to say a word about choosing a PhD advisor. This cartoon was actually found on a random desk in my lab, and someone wrote Yael ? Because at the beginning of your career, you see every person as a special and unique individual. I'd say after 5 years, I still see that. And with time, it's an amorphous crowd, then a bell curve, and then a data point. So I think when you choose your PhD advisor, you should try to look for someone who will be more of earlier in their career, so they can see you and they can tailor their mentorship and their project to you. Then after you have this really, key fundamental training, you can transition as a postdoc to someone that will just give you the space and the money to produce data and do it yourself. So that would be one of my advices when choosing a mentor. And again, one last comment that can turn into a whole discussion. I've now graduated one person, and I have two that are graduating this year and two at the beginning of next year. And I can tell you that another important aspect for me as a mentor is also to tailor the guidance or the career development of my people. Not everyone have to stay in academia. If you are, I'm very happy to support it. But different people have different ambitions and different goals. And I love this Oded Chavi's Twitter account, very clever memes. And one of these were really cracking me up, student to industry visiting his old lab mates. So my first student that just graduated started her job at McKinsey, and I'm very, very proud of her. She did an incredible job through her PhD. And my goal as a as an advisor is to train you to be the best scientist that you can, and then find your passion of how to apply it, in the outside world. Like I said, consulting, industry, academia, pharma, whatever that may be. So I don't think there are right or wrong paths. It's just a path that is good for you. And I was going to just put here in case you want to learn more about my lab and about epigenetics. So these are sort of podcasts we've done and interviews that depict some of our work. But I think I'll open the floor or the Zoom for questions. I have to thank incredible collaborators, and my lab has been very well support funded. And if you want to learn more about us, you're welcome to do it through social media or through one of our 2 websites, the official, the Sloan Kettering, boring one, or the one we actually care about. I think that's about half an hour.
Thank you